The support from the Ra Research Fund has been vital to me being able to create my own research program that focuses on ovarian cancer within the Centre for Cancer Biology in Adelaide. I would like to acknowledge that we have a wonderful collaborator in Professor Martin Oler, who's the Director of Gynecological Oncology here at the Royal Adelaide Hospital, and he's at the front line with the patients. So he's talking to the people who have ovarian cancer, and he actually does their surgery as well. I obviously have a wonderful mentor and very supportive lab head in Professor Stuart Pitson, who's the head of the Molecular Signalling Laboratory at the Centre for Cancer Biology. And for those of you who aren't aware of where that is, that's in the brand new building that's right next to the Morford Street Bridge. We have a facility in there which has a bunch of very dedicated, talented people that are completely committed to cancer biology. So that's a wonderful place to work, and it's a really wonderful supportive environment. So for 10 years, I've been working on a family of proteins called sphingosine kinases, which I'll just call SKs for short. And we've recently found that they contribute to ovarian cancer and are a new target for this disease. So before I got into this research, I wasn't really aware that ovarian cancer is the most lethal gynecological malignancy for women. And sadly, on average in Australia, 4 women are diagnosed with this disease every day. So the disease actually can start in one or both ovaries, and it can eventually spread and develop to fill parts of the abdominal cavity and kind of cover the organs in that space. So unfortunately, this makes this disease very difficult to treat because it means that it's already kind of metastasised, often by the time patients even recognise any symptoms. And so the surgeons then have to go in and undergo a lot of surgery to try and remove that tumour tissue from the abdominal area. This is then followed up with chemotherapy, and so most patients will have the chemotherapy that is the gold standard, which is paclitaxel and carboplatin. So while many patients will respond to this treatment, unfortunately, there are many women for which the disease will return within 6 months, and these patients are unfortunately called chemo-resistant patients, and so they now have to undergo more surgery and more treatment to treat the disease. And unfortunately, at the moment, the statistics are quite grim, and about 90% of those patients will succumb to the disease within 2 years. So this area really is crying out for more research and more treatments so those clinicians have more options to give their patients at this late stage. Right. So as a cancer researcher, I'm really interested in trying to understand why can some cancer cells respond to chemotherapy while others are unable to. They can just ignore the drug. And this is a similar question to why does a cancer cell start in the first place? What makes a normal cell turn into a cancer cell? So in this animation, which hopefully is going to work, this is basically just going to show you the process of cancer. So a normal cell is happily going about its job, but over a period of time, this cell might start to accumulate defects, which mean that it suddenly gains the ability at some point to become supercharged. And then this is just a cell that then can grow out of control. So it's a cell that's gone rogue. It's a cell that's undergone mutiny and it's just taken over. So you see that this cell is suddenly able to take over. It can grow, thrive and survive, and it might even migrate away from where it is. And that's, I guess, what we call metastasis. So the cancer cells have an Achilles heel in the fact that because they're growing so fast, they need a lot of resources. So they're sucking resources from around their environment, and this is basically how chemotherapy works. It targets those really fast-growing cells and hits them quite hard. And so many of those cancer cells will actually succumb to the chemotherapy and be killed off. However, there's in ovarian cancer and many other cancers as well, there might be some cells that are left behind, and those cells are left to regroup, and often they'll come back and grow back much more aggressively and faster than the original tumour. And so this process is something that we really need to target to develop new therapies. We need to understand, why is this process happening? What we're really doing is digging deep to try and understand the reason why these cancer cells don't respond to the chemotherapy, and we're trying to find that chink in the armour so that we can hit them and hit all those cancer cells so none of them can come back and take over again. All right, so one of the ways that cancer cells can survive the chemotherapy is that they are able to make their own signals to grow and thrive. So what we work on is a family of proteins called sphingosine kinases, and in normal cells these are normally tightly controlled. But in cancer cells they've been able to hijack this system, and so we have this hyperactivation, or the SK is really switched on, leaving the cancer cells to be drenched in a signal to grow and survive. So what we've found in ovarian cancer is not only is sphingosine kinase important, but it has a partner protein, SIB2. That SIB2 protein normally holds onto the sphingosine kinase and acts like a brake. So I guess if we're looking at a car analogy, the sphingosine kinase is kind of like an accelerator for cancer, and the SIB2 protein is kind of like a handbrake that holds onto that and stops the cancer cell from growing and allowing the cancer cells to be uncontrolled and growing like an accelerated version. But what we've found recently is that in ovarian cancer, the SIB2 protein's lost. So the cancer cells have lost their natural handbrake, and the sphingosine kinase is free to grow and accelerate cancer growth, cause cancer cell migration, and contribute to the chemotherapeutic resistance in these patients. So this makes sphingosine kinase a really important new target for this disease. So how can we go about tackling this? How can we go about hitting this system and finding a way to block this uncontrolled cell growth? Now, this is where we zoom in a little bit more on the target. So we're looking here, I know this looks like a giant blob, but we're looking here at a structure of the SK protein. So this protein here has many different surfaces, and what we have to do as researchers is work out what part of that molecule we need to hit. So we've done a lot of work to understand what is the best region of this protein to hit, and then try and find something that will bind to that and block its function. So there are many ways to go about this. One of the ways we can do this is to buy a mountain of small molecule inhibitors in and test them one by one in the laboratory. This costs an enormous amount of money, and it's something similar to trying to find a needle in a haystack. It takes a lot of time, it takes a lot of resources, and it's very difficult to do. So one of the ways that I've been able to speed up this process is to use high throughput computing technology to be able to make the computer do this process for us. So this is just an animation showing, which I should hopefully be able to hit, showing how this program would work. So basically we've done the research to show that there's this one part of the protein, this pocket here, that we need to hit the protein, to block its function. And the computer program will be taking, instead of in the laboratory doing this, it will take each of these chemicals, test them one by one, see if they fit into that pocket, and then from that we will be able to screen millions of compounds in a relatively short period of time. This saves a lot of time, a lot of money, and it allows us to be able to screen millions of compounds we wouldn't be able to afford and do in the laboratory. So importantly, once we've done this screening, we then get a short list of chemical compounds that we can buy in, and then we test those in the laboratory. And this is a wonderful way of doing drug discovery work. Of course, we then do extensive testing in the lab to make sure it hits this protein, it doesn't hit other proteins, and that it's actually effective at killing the cancer cells. And we've done a lot of work to prove that. So here we go, I've just got this, the picture here with the red, again that's that pocket that we've targeted. We have a small molecule inhibitor. It's kind of like finding the right key to a lock. We've gone through and we've screened a whole series of those locks, and it's kind of like the computer program allows us to take a huge library of keys and just have one tiny key ring that we actually buy and test. And maybe we test 20, 30, 50, depending on how many we need to test. So using this process, we've been able to find a small molecule inhibitor that hits the SK protein. And most importantly, we've been able to show that it's effective at killing the cancer cells, reducing the migration and metastasis of those, and that it's actually able to sensitize the cancer cells to chemotherapy. So I'll just show you one graph where we've looked at this. In this graph here, we see the white bar is our cancer cells. These are our control. The numbers are just how many cells are alive. So at the moment, we have 100% of the cells that are alive. If we treat them with chemotherapy, these cells aren't very responsive. So you'll see in the black bar, 100% of these cells are still alive. That's not what we want. We want them to die. However, if we take our inhibitor, which at small doses don't work, but if we combine these two, we suddenly see these cells start to respond, and in this short time course, that's really, really impressive. So we think this is a really exciting new avenue to undertake, and we're currently working up some really important, powerful preclinical models to be able to test this properly to show that this can work. We're also actively working on trying to improve our small molecule inhibitors so it's safe and effective to at some point go into patients. And so my ultimate goal is that this will go into phase 1 clinical trials. We have a lot of hard work ahead of us, but that is my ultimate aim. And my real goal in this is that at some point I can make an improvement on the 46% 5-year survival rate for ovarian cancer. I think it's not good enough, and we absolutely need more treatment options for the clinicians to give to those patients. So with that, I'd like to thank and acknowledge the Molecular Signalling Laboratory. You might recognise a few faces there. A few of them were in the video before. I'd like to acknowledge, of course, Professor Stuart Pitson, who's been an incredibly supportive mentor, and he's really helped me start my career and continue my career. This work is obviously a collaboration with Martin Oler and his team, as well as his research group at the University of Adelaide. He is a key person in this. He's our clinician, but he's also able to link us with the patients so we can get really precious tissue samples from those patients. That gives our work real clinical relevance because we get to test our inhibitors on patient material that makes it, really, really informative. I'd like to also acknowledge the Royal Adelaide Hospital Research Fund again for their incredible support over the years. I wouldn't be here without them. They have supported my salary. They've supported some of the funding to allow some of this work. This work has basically almost all been funded by this. And so, I'm really, really thankful for them choosing my applications. I'd also like to give a bit of a shout out to the research office. A few of you guys are out there, and I know you do all the hard yards in the background and get none of the thanks. So I'd like to thank you formally tonight for all the paperwork that you do. And I guess last but not least, I have to thank the donors for your incredibly generous donations. Every dollar counts. We put it all to good use, and I guess everything I've shown you tonight, you've helped to fund that work. So thank you for your incredible support.
Thanks very much, Melissa. So my name is Paul Reynolds. John did introduce me briefly. I'm chair of the RAH Research Committee, and just to give you a little bit more background on what we do, all of the donations that come through to the hospital are obviously greatly valued, and so any awards are adjudicated by a panel of scientists. So it's a competitive process, so we do our best to strive and ensure that the donated dollar goes to the best possible projects and the most promising young people. And as you've heard, Melissa has had at least 3 awards through our program so far, and she's just one of the many young scientists that we're hoping to promote. And ultimately, what we do find is that by giving them a bit of a head start like this and getting some runs on the board, they then become competitive for the really top dollar national awards, and that's what we hope to do, to actually use this program as a seed to continue to build for the entire precinct. So, Melissa, I've got just a couple of questions I might draw you out on your project before we sort of open it up to the general audience. With regard to the development of the little molecule that you are pursuing, I guess one of the things that struck me is that whilst you've shown us that you're able to use this to slow down the rate of the cancer cell in a tissue culture dish, so this is a little dish of liquid that the cancer cells are growing in, it's obviously a big step to get that into a person, and there are some hurdles that are needed to be overcome for that to take place. And so how do you see that would progress, where you can actually get a little product, whether it's a tablet or an infusion that might actually get into a person? Where do you go to do that?
That's exactly. So the funding that I've actually got this round is actually to test our small molecules a little bit further to make sure that they're actually going to be effective in preclinical models. First of all, we're also working really closely with the medicinal chemists, so we're working on improving that so we can make sure that it's safe and effective to take into patients eventually. There's a lot of work in optimising this, quite a few years ahead of making sure that we've improved it enough, and also that it's effective and safe and that there's not going to be any toxicity issues and things like that down the track. So I'm aware that there's plenty of years ahead of us testing and retesting and improving before it can get to phase 1.
Sure.
Yep.
And probably many people in the audience might already be aware that the usual chemotherapy we give for cancer treatment not only knocks the cancer cells, but it also knocks the whole body quite a fair bit, can cause problems with the immune system and people's hair fall out and so forth. With your approach, are you able at this early stage to predict what the sort of side effects and adverse effects might be? Do you think it would be safer than standard treatment?
I think it's probably going to be more targeted. So what we've shown is that in the laboratory, I know it's in a dish, but if we give these molecules to the cancer cells, they're very effectively good at killing the cancer cells, and they don't really seem to hit the normal. So that's it's a very artificial system, and obviously we'll have to test that down the track. But it seems like it's quite a targeted treatment so far. I guess the key thing is that also in cancer, the sphingosine kinase or the SK proteins are often hijacked, like I said. So they're at higher levels, they're switched on, whereas the normal cells are normally very tightly controlled. So it's, I guess you're kind of selecting towards something that will be hit hopefully more in the cancer cell than a normal cell.
Sure.
Yep.
And one of the other things about cancer treatment, again, people might know, is often there's combinations of drugs that are used, and I think you showed that in your slide where the combination worked better than the drug by itself. Where do you see that going with your particular strategy? Is there more than one molecule that you have that you might be able to combine to improve the effect?
So I definitely think, I guess, what we've found, in the ovarian cancer area is it seems like if you hit the sphingosine kinase like SK protein, you're able to sensitise the cancer to the chemo. So I think where we'd be heading is to see if we can combine our molecule, probably the lead molecule that we'll have, with the chemotherapy. Probably that will be the way we would go about it. And I do think it's worth us testing if it can be a standalone treatment, but I think the way forward is to target that first, because I guess those patients that don't respond to therapies really need other options. So I'd like to target that area. And I guess the fact that the CYP2 protein, that's that partner protein that holds the sphingosine kinase and stops it from, being uncontrolled, that appears to be in a proportion of ovarian cancer patients seems to be lost. So we have a real kind of biological reason for going after those patients that have chemo resistance. We might be able to screen them and say, these people seem to have lost their CYP2. They will probably respond, hopefully, to a SK inhibitor and work on that.
Right. And so I was going to ask about that. So do you see this pathway as being one that might allow us to improve our ability to detect ovarian cancer at an earlier stage? Because I think that's one of the problems with this disease. It often doesn't have many symptoms when it first starts up, and often by the time you get symptoms, it's sort of a little bit late to be able to completely remove all of the cancer with an operation. So if we had a marker that could be used to screen or detect this earlier, then do you think this is a pathway that could be used?
It's not something that I've got a research program around at the moment, but I'm definitely interested in looking at whether or not the SK could be an early prediction marker for whether or not, maybe if it's circulating levels of it, or if, it might be present in ascites in early stage. I don't know how far in you get ascites. Some patients obviously later than others, but I definitely think it's worth looking at because it does have this, I guess it's a target. We've shown that it's a target in this cancer. Yep.
And so just in terms of your own research program, how do you, as a evolving sort of mid-career scientist, and perhaps people aren't quite aware of the struggles that are involved, how do you, where do you sort of go from here in terms of seeking further funding as the years go on, and how does that work?
Yeah. Well, at the moment, I'm very much in the lovely support of Stuart. He's kind of got an umbrella over a bunch of us researchers, so the up-and-coming people are kind of pushing forward under his guise. I am very keen to, I guess, I'm going to be applying for a bunch of different sources of funding, and I guess there's commercial funding that I'm going to be going for to try and get support to then push some of this drug discovery stuff forward. So I've definitely got my eyes on a couple of grants that I want to push forward for that. But I think I'll be applying for the NHMRC, National Health and Medical Research Council, is the major funder, I guess, in Australia. It's incredibly competitive to get funding in that, but I do think that this body of work is lending itself towards going with that way. So if I can gain some larger funding and set this on a 3 to 5 year funding cycle, I can really push this work forward more quickly.
Yeah.
Yep.
And the National Health and Medical Research Council, as you mentioned, is the major national funding body for biomedical research, but the percentage of people who apply for salary grants is really very low. I think it's around 10% or sometimes less than that. So, and it's extremely competitive. So I think what Melissa's doing here is, getting obviously some good quality science, but also some runs on the board. So when she's up against the rest of the country, she stands a much better chance of actually continuing to progress. And so it helps to develop the whole precinct. There are a few questions that were submitted by some of the audience members. I'll just make a couple here. There were a couple that were related to consumer participation and health literacy, specifically Tessa Kowali,
I think it is.
how do researchers partner with consumers at different points of the process?
Right, so I guess I can speak for the research side of things rather than, I guess, at the hospital might be a different case, but at the Centre for Cancer Biology, we have a consumer advocacy group where we invite maybe patients that have had cancer or have cancer or their family members, and we invite them to come along and share their experience with us, and we talk to them about our research, and we very much involve them in our grant writing process and get them to read our grants and make sure that they think that the research that we're doing is relevant. It's an incredibly useful process because it puts a person's face to that disease that you're trying to cure, and it makes sure that you're asking really impactful scientific questions because you've got somebody right there. So it's a really useful experience, and we've gained a lot from that relationship with the patients, definitely.
We sort of covered a couple of things a little bit, but how long do you think it would take you to get to the next stage of your research? I guess that's sort of how long is a piece of string? But I guess that we must acknowledge that even some of the newer treatments that we read about in the newspapers now, I can well imagine 20, 25 years ago reading about antibodies and things, but there are some of these small molecules on the market already, and realistically, with success, how long would it take if everything went according to plan? Do you think you could get it into a trial?
Yeah, I think, given that we've got a really experienced team now of people who have done, drug development and drug optimization, we've got a really great medicinal chemist that's on board, and we should be able to, I guess, with the right resources and funding and time to actually do all the testing. I think I'm hopeful that within maybe 5 to 7 years we could be in phase 1 if all going well. I guess there's a lot of things that can go wrong along the way where things might get held up, but I'm hopeful that in about 5 to 7 years of working on this full speed, it might be able to be going into testing of safety in patients in the phase 1. So.
And it is the case that at the Royal Adelaide Hospital here, there is a phase 1 clinical trials unit, and there's also a commercial CMAX unit right on the same campus. So these things could potentially be progressed here.
I'd love to have Martin on board doing that, and maybe yourself.
Sure. Now, some of these questions are a little bit tangential, but Joe Clark had asked, can ovarian cancer be picked up as CT or CAT scans or CT scans? I guess that's probably more of a clinical question, and as a clinician I could say that ultrasound is a more sensitive way of picking up ovarian cancer. The CT scans give a cross-sectional view, and these little cancers often start off as very small spots, and so the CT scans are often used to determine just how far the cancer has spread. So they're good at picking up disseminated metastases like Melissa was talking about. But generally, I'm not a gynaecologist, but generally it is the ultrasound that is used primarily as the initial technique.